Okay, so we're here to talk about panel design in Flurochrome. So before that, let us talk about what do you understand by a panel? Anybody? So a panel is a combination of markers which will answer the question that you want it to answer. So it is a combination of antibodies. You tag it to different fluorochromes and you want to characterize your cells of interest. Now, the pattern is that there are some technological aspects and there are some biological aspects. Technological aspects is your instrument, your reagent, which fluorochrome, how to select that. And biology is what you want to assess. For example, if you want to do leukemia phenotyping, acute leukemia phenotyping versus a lymphoma phenotyping, your markers will be different, your approach will be different. And that is again something which we need to know upfront. So you learned about the instruments in the morning when Dr. Hemant spoke to you. So dependent on what instrument you will have a single tube or you will have a multiple tube for your analysis. So for example, if you have to do flow for BLL-MRD and you have only a 4-color machine, you might have to put up 5 tubes. If you have a 6-color machine, you might have to do 2 or 3 tubes. If you have an 8-color machine, 2 tubes, 10 color, 12 color, a single tube will do. So it depends on the capability of your equipment as well. Now, the number of colors that we can interrogate has increased exponentially. So when you looked at the earlier equipments, 3-color, 4-color caliber was something which many of us have used. And now we have easy equipments up to 12-color to 13-color available for routine clinical use, and the capability is only increasing. So in India, right now, there are labs which are using 16 or 17 colors, even in the clinical flow cytometry field. So with enhanced capability, you have an ability to take more markers, but your complexity of data and resolution and the data interpretation becomes more difficult. So our idea is that we should be able to maximize on the capacity of the equipment, yet not run into errors due to data interpretation. Okay. So the first step is that we determine the experimental goal and approach. The next part is knowing the biology. What you want to do, how you want to do, how are the antigens expressed in your cell is important. Then key is knowing your flow cytometer. So you know which detectors your flow cytometer has, which lasers it has, so that you can select the correct channels and the correct fluorochromes. The next step is to assign the fluorochromes. Assign the fluorochromes. The key point is that when you are choosing markers, for example, you have an 8-color experiment, you have 8 markers which you want to use. How will you tag them to different fluorochromes is determined by matching it to the brightness. So if there is a dimly expressed antigen, you want to give it a boost. So you can conjugate it to a bright fluorochrome and vice versa as well. Important is that if you've decided that these are the 8 markers, these are the 8 conjugates that you're going to work with, you have to first review the theoretical design, understand if there are any issues, any problems that you're going to run into, and then you have to optimize. Optimizing means that you order the reagents, you start running the panel, and then you see what kind of problems are coming up. And if you see the blue arrows on the right that have always been there, so what you are seeing is that even after running a panel, it doesn't mean it is perfect. At any step of the panel design, if you feel that there is something wrong, if your data doesn't look good, or if you see potentials of error, you have to go back to the previous step. This is called iteration. So understand that if we order a panel, it may not be the perfect panel and it may not work perfectly in our lab continuously over a period of time. So when we start it, we have to ensure that, if the data doesn't look good, go back to the previous step, do the titrations or do the assignment of fluorochromes again. Sometimes you may have ordered an antibody, say cytoplasmic CD3, you ordered in a dim fluorochrome such as PAC blue. And now you feel that your bright population versus your negative population you're able to resolve. But in an acute leukemia, what do you want? You also want to capture the blast, which will have 50% of the normal expression. So you might have to tag cytoplasmic 3 to a brighter fluorochrome. So what works in one setting, in one equipment may not work on the others as well. So the first component is knowing your population of interest. So we need to know what is the objective of the panel that we are designing. So acute leukemia, as I said, will be a different panel. B-cell lymphoma will be a different panel. The objective needs to be decided first. Then the next question is, what are your populations of interest? So if we take an example, for those of you who are interested in leukemias, for example, or lymphomas, for example. So for a B cell, for a lymphoma screening tube, what is your objective? To identify the B cells, to identify the T cells, the NK cells, look at the basic CD4, 8 subsets, see whether there are any gross double positive, double negative populations. For a B cell, you would want to do a clonality assessment, at least assess a CD20 on the cells as well. So with all this, you know what kind of markers you want and what are the populations that you are going to use. At this time, it is also a good idea to have a subtle gating strategy in your mind. So, okay, 19 versus 3 to separate B from T. Then among the non-B cells, maybe use 3 and 56 to separate your T cells from NK cells completely, and also identify the NK T cells, which will express 3 and 56. So this kind of a basic gating strategy should be in your mind. So now simultaneously, you know that when you talk about 4 and 8, so cells which are 3 positive will express 4, which means 3 and 4 will be co-expressed. If you talk about 8 positive cells, again, it will be a 3 and 8 co-expression. So you also have an idea about which markers are usually co-expressed. Now, the next question is what tissue you're going to use. So the fluorescent backgrounds, the signals, they vary dependent on the tissue. So if you are doing a lot of mechanical dissociation on a tissue, the assay will be different. So for intracellular or surface markers, again, there are key different issues. For intracellular markers, it is important because they have a lot of high autofluorescence that is introduced once you do the permeabilizing step. Now, the next step is stimulation. For those of you who want to assess cytokines or increase in the, or maybe for PID, if we say PMA stimulation and you want to see the DHR emission in the granulocytes, you'll always see that in the stimulated, the unstained cells will also move. So that also is a consideration that you need to have. A practical consideration is whether you're planning a panel as a short-term or a long-term panel. If it is a 10-color tube, it is going to be more complex than a 6-color tube. So that you have to keep in mind. So when we want to select markers, we have to decide how many do we want to answer the question. So for the B-cell lymphoma example or the lymphoma example that I gave you, screening tube, what do you think is the minimum markers that you would want? So 19 B cells, 3 T cells, 4, 8 T basic subsets, kappa lambda. So we are at clonality. You want 45 for gating. Along with that, now is the question, what would you want? So if you want to assess NK cells, 56, you'll add to the mix. If you want to look at TCR gamma delta, you'll add that to the mix. Then if you want to see plasma cells also in the same equation, then 38 to the mix. So you get a list of markers that you want. And now dependent on what populations you want to record, how many colors you have, you will decide how to spread it. So when we talk about marker expressions, there are 3 different kinds of expressions. One is where you have primary antigens. Primary antigens are brightly expressed antigens. So when Kunal was talking to you, he was showing you CD4. It is a very brightly expressed antigen on a normal T cell, right? So you know that 4 expression in the normal T cells is bright, in the monocytes it is moderate, and it is negative in the neutrophils. So these are called as primary antigens. Usually they define lineage. Another example is CD3. So CD3 defines a T lymphocyte. It will be absent in all the other populations in the peripheral blood, right? Now, secondary antigens are those which are expressed over a continuum. So over a continuum means that cells may have very low, then moderate, and then slightly short of bright, you will have signals. And tertiary antigens, they are the ones which are critical markers for those who are assessing T regulatory cells, for example. So CD127 becomes the marker to identify T cells. It is expressed at a low intensity, so it becomes a critical marker. We have to know which markers are co-expressed. So try and make a mental picture of what is co-expressed. Again, identify the gating strategy. And for clones, it is always important to choose the right clone. For example, if you want to assess the MDS, so you want to assess CD16 on the granulocytes, you want a clone of CD16 which will react well with neutrophils. You don't want a clone of CD16 which you may have a clone of CD16 which works very well with NK cells, but it may not react to granulocytes at all. So you can get that information using published literature, sometimes asking your friends who are doing that experiment. So as I said, we've already classified this. So based on the lineage, these are examples. So CD3 for T lymphocytes, lineage defining, not expressed in other cells. 19 for B cells in the context of lymphomas, definitely lineage defining. Then CD56, again for NK cells. CD4 is good for helper cells. Secondary antigens are expressed at high intensities and over a continuum. Tertiary antigens, you have low level expression. And for clone and gating strategy, again, it is according to the published literature, according to your experience as well. And trust me, if you gate something a certain way today, will not be the exact way you use 5 years down the line when you gain more experience with the assay. So once you know all this, you can also do one thing, that if you have 8 markers on your list, see what fluorochromes they are available in. So there might be a marker which is conjugated with only 2 or 3 fluorochromes, and that will limit your options of selection. So you should know that information beforehand. And there are multiple websites, for example, Compare Fluorofinder, where you can make accounts where you don't have to pay and you can just see antibodies across multiple vendors. So earlier what we would do is, if we have to order antibodies, we would go to vendor A's website, and then we would see what clones they have. Then vendor B's website, note down the clone. So these websites, they make your life easy. And the note especially has to be made with markers which are available with rare fluorochromes. So nowadays, the availability of fluorochrome conjugated antibodies is far easier than the earlier days. But still, for newer markers, such as, for example, when you talk about CD319, you know that you want a bright fluorochrome with 319 in plasma cells. So for those antibodies, we definitely need to keep that channel free. For gating strategy, this is an example. This is for an acute leukemia gating strategy. You want a 45 versus side scatter gating. In addition, you would want to have 34 maybe in all your tubes to guide your gating strategy. Now, this is the example that I was giving you, and this is something that I made a mistake in the lab in. So what we wanted to do was, we had CD16 antibody lying around with us, would react very well with NK cells, was being used in the lymphoma panels. So we started to do the MRD assay, picked up everything that was in our fridge, conjugated that 16, used that 16 antibody, and our data looked like this. So if you see no signal in 16 when we look at the myeloid cells. So this was one particular clone. Assessing it with the other clone, you realize that the signals were always there. So in the middle, you have the normal samples, and you see that the 13 versus 11B plot is the same, but now the CD16 signal is coming up. So it is important to have the right clone. And this is something that I learned by making a mistake. Now, the next component of your equipment or a panel is your flow cytometer. So what is the laser and filter combination? Dr. Hemant showed you those in the morning. You need to know whether you have a 2 laser, 3 laser, 4 laser, 5 laser equipment. And you also would like to know how many colors can each laser detect, and what are the exact bandpass and other filter combinations that are available. So there are 2 things here: the laser wavelength, detectors per laser, and the filters. So for example, these are 2 examples. The above one is from a lyric, and you can see that you have blue, red, and violet laser. The bottom example is from a Cytoflex, and again, you have 13 markers. You again have the same 3 lasers, but the detectors, they are not the same. So for a Beckman equipment, okay. So do I have a pointer here somewhere? Okay. Cursor के साथ the screen sharing comes up. That is a problem. Okay. So in any case, so we know that Beckman laser on the blue laser on the Beckman equipment, you have 5 channels instead of 4. So the other channel, the third channel that is there, is ECD. Now ECD uses a 610 by 20 bandpass filter, which you cannot run on a lyric, because the lyric doesn't have the same bandpass filter. It doesn't have the same detector there. So this is something which you need to know. For example, if you see a published 12 color panel in which ECD dye is used, you cannot extrapolate that to your instrument if your instrument is a lyric, right? So you need to be aware of the combinations of the filters and the lasers available in your equipment. Now, next comes the component of fluorochrome selection, and we'll spend more time on this in my subsequent slides. The aim of selecting fluorochromes is to ensure that all your markers, they are expressed at the level that you want to see them, and they don't spill over into the other channels. The laser instrument and filters will decide what fluorochromes you can use. The ability and the other component, as I mentioned earlier, also was whether your required marker is available in that particular fluorochrome or not. There are tools which Dr. Hemant also referred to. Every company has these tools where they give you an offer of building a panel for you. And there are three important things which we need to remember. We'll come over that in the subsequent slides. One is cross laser excitation. For example, if you have uh, uh, a dye which is excited by the blue laser and it also gets excited by the red laser, then it will start emitting something in the red laser as well. So that we need to keep in mind. For fluorochrome spillover, again, compensation Kunal has referred to. Compensation in the current era, we do usually by the beads or by the cells in the automated instrument-based compensation. It is very good, but it is not perfect. So we have to do some post-acquisition compensation as well. And for pairing of antigens with respect to the intensity expected, always remember dim antigen, bright dye, bright antigen, dim dyes. And for co-express markers, especially the ones which are co-expressed at a dim intensity, the spread of the other marker will matter. So here I'm showing you an example where you can see that PE Texas Red and PE 6CF594 both have the same excitation and emission properties. So obviously these two dyes cannot be combined in an experiment. Now the same thing is true for PerCP or PerCP Psi 5.5. If you see, a large part of their emission spectrum will overlap. So these dyes, they should not be combined together in an experiment. Now same thing for APC and Alexa Fluor 647. You can choose either of them, but not both in an experiment. The same thing is true for BV421 and the V450, as well as, so these two dyes you cannot combine in an experiment. So this is basic, simple thing. Now, once you get your panel, you titrate your antibodies. Titration Kunal has shown you. You have to run controls. So compensation controls he referred to. In addition to that, the unstained tube acts as a very good control because it gives you the amount of background fluorescence that your cells with that particular processing have. In addition to that, something called as fluorescence -1, and I'll tell you that in a little bit of time. So once your panel is done, once you've ordered your antibodies, don't try to pipette each antibody in each tube. Always prepare a cocktail. A cocktail means that maybe for 10 tests, you multiply each antibody's requirement by 10. So titrated volume of the first marker was 2 microliters multiplied by 10, that comes to 20, so on and so forth. And then you mix it. And you use the overall requirement. So 1, 2, 3, 4, 5, 6, 7, 8, 8 markers, add the requirement. So out of the cocktail, take that much when you are staining your cells. So cocktailing leads to lesser pipetting errors, and you have to validate your cocktail. Very important to make adjustments in your panel if there is some error. So now keep in mind that a bright fluorochrome will emit more photons per unit time. And this is the concept of brightness of different fluorochromes. So FITC, relatively dim dye, 0.5 was the molar extinction coefficient, while for PE, it is a bright dye and it will lead to more photons. So these are the two things which are used. Molar extinction measures how well a fluorochrome absorbs energy. Quantum yield is once it has absorbed energy, how much energy it releases. So brightness is a function of these two. However, it is also dependent on the instrument and the particular laser concerned. For example, so therefore, brightness is a function of the fluorochrome instrument as well as the laser. So in test conditions, Kunal showed you an example where he showed you something where you had populations stacked and there was some negative signal which was wider and the bright signal was at a same point. So if you look at these two scenarios, what you see is that the signal, bright signal stays the same. The negative signal median also stays the same, but the width is widened. So if you have a population that should be present here, that is between these two peaks, which of the plots do you think will resolve it better? The left or the right? The left one, right? Because there is more space for that dim population to form a distinct peak. So this is the mathematical calculation that is done. So you calculate the difference between the median of the positive to median of the negative, and you divide it by the robust ST of the negative population to obtain the stain index. And this is good because in your sample, if you are working with an antibody, you can calculate the stain index of that titrated volume to know how much area you have for separation of the bright and the negative. So this is also a measure of your reagent on your cytometer once you've normalized for everything. So this is an example where the PE signal, you have the stain index calculated, and these values you can easily get from your cytometer. These are the stats which your cytometer will give you. So you have dyes of varying brightness, and these are examples from 3 different vendors. So on the left, you have an example from Beckman Coulter, and you can see that you have varying brightness. So bright dyes, P, PC 5.5, PC 7, these are your bright dyes. And this is also variable upon the laser. So when I was saying that if a particular dye gets excited by multiple lasers, its brightness, for example, for PE, is lesser with the blue laser versus the yellow laser. So if you have the option of yellow laser on your equipment, you can choose what you want to do with the detectors that are common to both. On the BD equipment, again, you have very bright dyes and they are given across all lasers. Then you have bright, moderate, and dim dyes. So what would you like to do? If there is an antigen which is strongly expressed, you would use the dim dyes, any of the dyes in this column, dependent on the laser that you have. While if you have an antigen which is which is usually dimly expressed, you would like to conjugate it to a bright fluorochrome. Again, across lasers, you have these options given. And on the right, you have this data from Thermo Fisher. So you can choose which of these you want to use. Now, the next component that we referred to earlier was the antigen density. So for your normal cells, what is the level of the antigen that you want to interrogate that is present on the cell is a measure of the antigen density. So for that, we can harness the information that is already available in the published literature, sometimes on the manufacturer website. So on the right, you have 2 examples from manufacturer websites where they have given you the marker expression for normal T cells. So you definitely know from this data that 4, 8, they are bright. When you compare it to, say, CD27, 28, and then 25 is a very dimly expressed antigen. In addition to that, your past experience will help, and you can determine it yourself using cal particles, but we are not going into the detail of that today. So compensation Kunal referred to. So FITC spilling into the PE detector. This is like P would say, I don't want FITC in my house. Why is it entering my house? Sometimes your kids do that, when when their friends come over who they don't like. So P would think that I wish FITC would rather stay in its own house. So when you see compensation, you are basically removing the FITC signal in the PE. Here, PE doesn't have any staining. This is only a FITC stain tube. So you want to remove the FITC signal spilling over into PE, and you're able to remove it, and you get them to the same median. So ensure that you don't do eyeballing compensation. Ideally, in a comp experiment, we should be looking at these medians to correct the compensation. So this is 16.4% of the FITC of the PE signal, which needs to be subtracted from the PE, and this is the figure that your instrument will have. Why do we want to compensate? Because it impacts our data quality. If you're measuring quantities, it will impact our quantities. So if we look at this is the correct compensation. You have 21% of 5 positive B cells. If your data is undercompensated, the number increases. If your data is overcompensated, the number reduces. More often than that, you might miss dim expressions if you have overcompensated data. So what are the sources of these spillover? The spillover happens in adjacent detector. One example was FITC spilling into PE. Now, the second example is similar emission spectra across the different lasers. For example, PE Psi7 and APC H7, they are excited by different lasers. So PE Psi7 by the blue laser and APC Psi7 or APC H7 by the red laser, but they emit in the exact same filter zone. So they also have some amount of spillover which needs to be corrected. And the last one is residual based fluorescence. For example, if you're using tandem dyes, so PE Psi7. So the PE Psi7 dye, the PE component of it will also lead to some fluorescence in the PE channel. So these are examples of these 3 different phenomena. Keep in mind that there are issues that will happen. So BV421 will spill into 510 and you have to correct for that. And similarly here, PE Psi5 into APC is a common problem. And the same thing that I told you for PE Psi7 would exist for every tandem dye. So for APC H7, it will spill into APC, and a BV711 will spill into BV421, which it is a tandem of. Now, Kunal referred to something called as spread. So spread You can correct the amount of signal spilling into PE for FIT-C by doing compensation, but you cannot correct the spread. So if you look at this example of PERCP versus PE size 7, you see that if this is the badly compensated data, this is the robust ST, you correct it, you compensate it correctly, you bring the cell down, so your negative MFI will match. So your compensation is perfect now, but the robust ST of this population does not change, which means that now this population, which was earlier a compact population, has actually become broader. So that is called as spread. So if you look at the PE size 7 here, the dim signal of any marker in this zone will be very difficult to distinguish from the positive signal of PERCP. So this is what is called as spread. Now, what spread leads to is if you have two markers here, you have a marker B on PE and say a marker A on PE CF594. So you have spread of population A versus B. Now, A does not express B, so there is a lot of space here, right? So a huge amount of space, good stain index. Now, if you have A, which also co-expresses B, and B is expressed on the positive population, which is isolated B positive, only B positive population at a high intensity, what happens? This space reduces, right? So from a single positive where A versus negative for A, you could resolve very, very well. The A positive, B positive becomes difficult to resolve from B positive because B positive cells, they have a wide spread. So this is something that we need to keep in mind because this leads to loss of resolution. So the amount of spread is related to the spillover and to the reagent brightness. And reagent brightness is a function of the antigen density and for fluorochrome brightness. So as you will increase the colors, there will be more spillover and there will be greater spread. So a key point to also remember is the co-expression of markers. So marker A and B, ask yourself if they are co-expressed. If they are not, you don't have to worry. You can assign the fluorochrome based on antigen density. However, if the markers are co-expressed, think about which marker is more than the other. So if A is more than B, then you use a bright marker for B, use a relatively dim marker for A. And you'd use a marker that should not affect the resolution of the single A positive population because you don't want a population, you don't want a marker which is too dim so that you cannot now resolve A positive from A negative as well. And these are the resolution impact matrices which many companies will give you. And the red areas are the overlaps which you should avoid for co-expressed markers. This is for a lyric instrument. And again, the red areas, they highlight the parts that you should not use markers that are co-expressed in these two detectors. So for example, you would not want to use PERCP with PE on a co-expressed marker if you can. The other things that affect resolution are also how you process your sample. For example, the fixation that you're using, the permeabilizing step that you're going to do, how you're going to stimulate cells, and what buffers you have. If you have many brilliant violet dyes together, they may have errors in compensation because you need a buffer to stabilize it. Thank you. Now, additional markers that need to be considered for any intracellular staining is that intracellular staining will always have high backgrounds because of higher autofluorescence, because of the fluorochrome to pass through cell membranes, because of permeabilization, and the washing steps, they are critical to keep low backgrounds. The other key point is if you have CD3, say in any fluorochrome, and you are using 5 microliter for your surface titration, that volume may not work for your cytoplasmic experiment. So if you want to change the way you use an antibody, you have to always titrate it again. So titration, we have already seen Kunal talk about that. This is an example of an experiment for CD20. And this is the kind of graph that you can plot. On one axis is the signal noise ratio, which you saw earlier when Kunal was talking to you. However, there is another entity called as the stain index, and this we referred to earlier. So you can calculate both and choose the best option, choose the best concentration of antibody that gives you a best match of signal stain index and the signal noise ratio. For example, for this antibody, it is the concentration of 3 microliter that gives us both values at the highest. For cytoplasmic 3, as I showed you, again, for cytoplasmic markers, always when you're doing the titration, use the same processing steps as you're going to use when you are going to process your samples. And for running controls, you have to use instrument QC. So for example, if you have a BD equipment, a CSNT bead for Beckman, it will be different. For fluorochrome controls, you do the compensation controls. In addition to that, once you've designed your panel, ordered your reagents as a single practice, one go in the beginning, it is good to do FMO controls. FMO controls will account for spread, fluorescence -1. So basically, if you have 6 markers, add 5, remove 1. Then make the next tube, add the other 5, remove 1. So if you have 6 experiments, if you have 6 tubes, you will have to, if you have 6 colors, you have to make 6 tubes for fluorescence -1. And then you have your assay control with autofluorescence, negative and positive controls. And these give you a good guide to assessing it. So fluorescence -1 controls I've referred to. So in a practical panel design, first key is you should know what you want to achieve in an experiment. What is the goal, whether it's a simple versus complex experiment, short versus long term, whether you're going to use surface cytoplasmic staining, nuclear staining, whether you're going to stimulate your cells or not. You have to understand the biology of your cells as well as the antigen expression, whether your antigen is rare or it is a widespread antigen, what is the density of the antigen. You have to know your flow cytometer. Without that, you will never be able to get to a good panel. Then comes the component of maximizing resolution. Assign fluorochrome according to the brightness of the antigen, and co-expression always needs to be kept in mind. The key is to reduce spillover, to reduce the spreads. And once you have a panel, you always test, iterate, and go back to any of the earlier steps if it does not work well. So with that, I would like to end and acknowledge Dr. Paresh, my colleagues. So Ganesh is here in this picture and Richa in this picture. Along with that, Dr. Himil, who we have asked to run a lot of these experiments. So thank you so much. And I'm happy to take questions if you have any.